Good afternoon. So, welcome to this session of current status and trends in the analysis and quality control of small molecules, biologics, and biosimilars. We have one talk in small molecules. You're going to talk about small molecules as well. And the rest of the talk is in large molecules. So we have a really good session waiting for us. Without further ado, I would like to introduce Rosario Lobrutto from TEVA, and the title of his presentation is Analytical QBD, Method Inception to Method Transfers.
All right, welcome everyone. Thanks for the kind introduction and the invitation to speak at Pittcon this year. My talk is going to be on analytical QBD, method inception, method transfer. Whether it's small molecules or peptides, generally the same principles apply. There could be some specifics, additional specifics for peptides, but overall the same concepts apply. So what is the goal for analytical QBD? Analytical QBD goal is science and risk-based. So basically, at the end of the day, for analytical QBD, you want to design a method that consistently delivers the control of the quality attributes of the drug product, whatever that might be. That could be a transdermal dosage form, a parenteral dosage form, a tablet capsule. And QBD is performed and product process is performed to understand the product and the process understanding. Basically, it's the eyes into the process. We need to know what the QTPP is, quality target product profile. The CQAs, it's the critical quality attributes of the dosage form itself. The CPPs, which are the critical process parameters of the particular process used to make the more formulation or drug product, and the control strategy for each of those CPPs. This all revolves around the drug product itself. But now when we start talking about the the analytical method, we also have things called CMAs, critical method attributes. We also have other things in terms of CMVs, critical method variables. And we also have a control strategy as well too. What I'll try to do is delineate those terms and to give you some examples and background supporting what a CMA might be, what a critical method variable might be, and how to control it. So if you take a look at the drug product design space and the analytical profile design space, they need to overlap. So you're developing a formulation under quality by design. The formulation you develop needs to be robust, but it has a certain design space. You need to ensure that your method is able to extract the same amount of drug, whether they make it with a slightly modified process A or process B. We need to determine that the method is still selective. We need to determine if we can still demonstrate appropriate accuracy and robustness. So you have to understand how the formulation variability is controlled by the method capabilities. Because a lot of times what might happen is you develop a formulation under the QBD approach, then you have your target formulation, and then you do all of your method validation on that target formulation. Or even you're doing analytical QBD and you're doing these DOEs and design of experiments, but they're all done on the target formulation. But that's not necessarily the case, because if you're filing with a design space, you're going to be having a formulation that could be made somewhere within that design space. So your method should be able to control the dosage form for the particular quality attributes. All right, another thing in quality by design is a few elements. One is knowledge management. The other one is risk management. Knowledge management is all the knowledge that one would gain from method inception all the way to transfer and continual improvement. There are a variety of softwares out there that help you with the knowledge management. That's not the purpose of this talk. However, it is encouraged to have some type of systematic way to retain the knowledge, leverage on the knowledge. As you can imagine right now in this very dynamic pharmaceutical world, different laboratories might be working on the same product. Products are being transferred from one laboratory to the next. CMOs or CROs are assisting with some of the work. How to basically put all that knowledge into one bubble and retain that knowledge and leverage on it in the future is extremely important. But let's go back to risk management. So with risk management and with the advent of quality by design, it's not, oh, I have a fire, now let me go find the fire hose and put it out. As opposed to proactive, basically, risk management, before there's a fire, well, what are the different ways we could put out the fire? Where is my fire hose? Where is the fire extinguisher? Do we have an evacuation plan? Who do we call? So that's proactive risk management. So then by the time, if a fire does come about, everyone's calm and reserved and could put the fire out in an easier way. As opposed to the reactive one, the job will get done, but at a certain expense, and quality could be compromised. So we want to move from a paradigm shift of reactive troubleshooting to proactive failure reduction, prevention of failures, communication of risks, team-wide understanding of critical risks at the local level, with your customers, and also with your management. And then risk acceptance by management. There's no way that you'll ever be able to eliminate all risk. There's going to be some residual risk. How much residual risk will be tolerated or accepted is something that needs to be discussed, and that's within the company infrastructure. Sometimes to get rid of 99% risk might take X time, but to get rid of that 1 extra percent might take many, many, many more years. But you have to look at not only the time it takes, but the impact it has on the quality of the product, because the safety of the patient is the utmost importance, right? So safety and efficacy. So how does the risk impact the safety and efficacy of the product? Now, as you all may know, there's ICHQ-9, which talks about quality risk management. Each company has their own interpretation of ICHQ-9. This is just my interpretation of ICHQ-9. In the beginning, a project team is generally formed. You enter the risk identification phase. In the risk identification phase, you typically have a brainstorming session. This is where you brainstorm about the method. What are the potential things that might go wrong? What are the things that we might need to control? You gather information about the physical chemical properties of the analyte, knowledge about the excipients, potential interactions between the excipients and the API. If you're working in the generic world, there's a lot of information in the literature there. If you're on the branded side, that information you might have to leverage on some information from similar molecules within the portfolio or pipeline. You then do some method variable mapping. I want to use the word variable. We're moving away from the term factor and parameter. So the word we tend to use now is variable. So you'll start to see that a lot more often in some publications and presentations, and I believe the USP might be also adopting this verbiage in the future as well. Then you have a risk analysis, and there's a variety of risk analysis. One of them, typically what you do early in development, is fishbone risk analysis, and traffic tool, that's a heat map. I'll show some examples of that, and then the FMEA tool, which is the failure mode effect analysis tool. Then eventually what you end up doing is you're doing some type of DOE design. Now DOE stands for design of experiments. So basically here you want to do the right amount of work at the right time. It's not design on everything, okay? It's design of experiments. Sometimes when statisticians get involved, they get very excited and you're generating a lot of data, a lot of experiments, but is it really value added? You have to truly define and do your homework up front as a team to ensure what variables might truly be critical and what impact may they have on the method attributes. So you may have a preliminary DOE for screening, and then later on, when the formulation gets fine-tuned, or if you're on the API side, when the synthetic process gets more or less locked, then you can do an optimization DOE. You may not want to do such a detailed DOE up front because the formulation can change. Remember, the formulation development is dictated by multiple things. It can be dictated by a clinical study. It can be dictated by the stability. And also can also be dictated sometimes by the manufacturing process and the cost and the operations capabilities as well too. So you need to take all those things into consideration because that's what drives at the end the formulation development. We then move to the project team, and this is where the project team, they peer review the risks with other experts. Remember, it's a team. There's no I and we, right? So it's a team. So R&D works with QC or quality control because your method's eventually going to be transferred there. So one of the benefits of working together much earlier with QC is having them involved already before you even start validating the method. You may say to yourself, well, due to project attrition, the project may never reach QC, but you have to take a look at your overall attrition rates. Just say they're 70%. Well, you know what? It might make sense to have QC involved because, you know what? The method's going there. You can gain some variable input from them. What types of equipment do they have? The experience level. Is there special detectors? Do they need to get special training? Is there special equipment where they need to order it as part of their CapEx? What's the lead time? All those type of things play a huge role into ensuring a successful method transfer. Later on, when the team discusses, and by team I also mean not R&D and QC, but also regulatory as well as the quality group are all involved in this, under some good facilitation risk management, there's a management review of the risks. So the team presents the risks. Here's our residual risks. This is why we believe there's residual risk. It's a low impact in terms of impacting safety and efficacy. And the management team would need to accept those risks or not. The next thing is the project team that defines a control strategy and the recommendation. So for anything that's critical, like you have a critical method variable, which is impacting a critical method attribute, you would put some type of control strategy. Control strategy could be additional description in the method in order for you to, let's say, pay specific attention to the pH or to the temperature, or maybe a special way to do integration. Integration is actually a very important point because you're not supposed to integrate differently your release and stability results compared to your validation. So the way that you did your validation, you need to use the same integration routine for your release and your stability. And that's something that typically you may want to put in your method, especially for some challenging separations where sometimes you may do a tangential skim, sometimes you may drop the perpendicular. Whatever it is, you need to justify your specifications. And at the same time, you also need to justify why you're integrated in a certain way. And the last thing is after you identified all the risks and you presented them, looked at the residual risks, it's up to management to challenge them to accept the residual risk or not, and what is the risk mitigation plan. So I briefly talked about a few steps, which was the brainstorming method variable mapping, the cause and effect diagrams of the fishbone diagrams. I'll show you some examples of that. Then the heat map, basically it's like the traffic light risk analysis, red being high risk, green being low risk, and yellow somewhere in the middle. After that, you would go ahead and do a DOE1, I call it, a screening type of DOE. Generally here you might have several variables, 4, 5, 6, and you may not want to do a full factorial DOE, so you do some type of partial factorial DOE or a Plackett-Burnham design where using like 11, 12 experiments, you can screen a multitude of variables. Later on, after the formulation process gets locked or the API process gets locked, if you're on the API side of things, you would then do a more formal type of risk analysis, which is a failure mode effect analysis. And then after that, you would do an optimization DOE. At this time, the number of variables have probably been narrowed down to a few of them, 2, 3, or 4. Here, you could do a full factorial with some replicate analysis and then possibly a partial factorial if you still had a high number of variables. And then based on the outcome of that DOE, you would go ahead and update your FMEA and then have the appropriate control strategy. I'll emphasize these points with examples in the rest of the talk. So what would a brainstorming session look like? So you have an initial separation. You did some type of gradient screening, and you have a critical pair. And then you as a team then discuss, all right, what could be the things that could impact that critical pair? And of course, you have to take a look at the structure, if you know what that impurity is. On the API side, you may have more knowledge of that because it could be a synthetic byproduct, as opposed to on the drug product side where it could be more a degradation product. But in any event, you would say, all right, it could potentially it's ionizable. We did some initial pH screen. We saw the main peak and the impurity both moving. So that means it's ionizable because it changes the function of pH. Could it be sensitive to temperature? Could it be sensitive to the buffer concentration in the mobile phase? Maybe it's a peptide, and here ionic strength plays a much bigger role. Organic modifier concentration in the mobile phase, the gradient slope. So this is kind of just a brainstorming exercise. It doesn't take that long. You get together a couple people in the lab who are working on this project or from other projects, and you kind of write this down. Once again, part of knowledge management, you have to capture it somewhere. Then what you want to do is you construct a fishbone diagram. At the head of the fishbone is your critical method attributes, sometimes called CMAs. On the left-hand side, which are the causes, these are basically the bones. This is an example from HPLC, for example. So you have a buffer solution, you have mobile phase composition, you have temperature, gradient, injection, and detection. There's no right or wrong way to do these fish bones, but you really got, the only thing you need to consider is the causes go on the left and the effects always go on the right. So if you change something on the left-hand side, how can it impact something at the head of the fishbone? Then what you can do is you do a heat map. Heat map is something very simple. Remember, if you want people to start using quality by design, you can't be putting too many rules in place at the beginning. You have to have people feel comfortable, right? So you have them start using a traffic light risk analysis, green, yellow, red. And you can see the critical, or the method variables are on the x-axis, and on the y-axis you have the critical method attributes for this particular method. You would discuss as a team, could, for example, column temperature impact the resolution, yes or no? Could mobile phase buffer concentration impact the resolution, yes or no? And typically, for anything that's yellow or red, you want to write down a few notes about that. Now, as I mentioned to you before, there's a lot of great software out there that allows you to capture this, and it's version controlled, so at every iteration you can do that. But in the infancy stage, if you want to just use an Excel file and capture notes in there too, that's something that could be used. All right, so let's talk a little bit about critical method variables and critical method attributes. So in the top example here, so you have a gradient, and here it's a steep gradient, and then you have a certain resolution of critical pair. In this particular case, you vary the gradient, it's a shallower gradient, and the resolution changes. Now, not all the times that you change your gradient slope does your resolution change. In this particular case, since the gradient slope was changed, we saw an impact on the resolution. That means this variable here, gradient, is considered a critical method variable. So let's talk a little bit more about critical method attribute. It's an element of method performance that must be measured to assess whether the method is capable for producing fit-for-purpose data. Generally, it's part of your system suitability. Your system suitability criteria shouldn't just be what a copy-paste of prior methods from before, right? There should be some science behind it. How did you come up with those system suitability criteria? It should be scientifically driven. Another point is one method variable may impact multiple CMAs. So for example, if I had gradient, it's possible it could impact resolution. It could impact tailing factor. It could impact retention time. Also, multiple method variables may impact a particular CMA. So, for example, gradient could impact the resolution. The pH could impact the resolution. The temperature could impact the resolution. So for any variable that is deemed to be a CMV, or critical method variable, you need a control strategy. This control strategy ensures quality systems are in place for the method and system suitability for control of high-risk variables. Point of note here, control strategy should be something that's in place that catches a failure of the critical method variable before injection of your sample, okay? Because if your sample gets injected, then you have an LIR, and then you have investigation, and then you have all the follow-up items that happen after that. So you have to have something in your method, something that you can see, something that you can detect before the injection of your sample. Okay, that's what a true control strategy is. So let's go a little bit back into the critical analytical method variables. In this example, you have pH of buffer as a method variable. Critical method attribute is resolution of critical pair. Is the critical method variable critical, yes or no? If the pH of the buffer has an impact on the resolution, in this case, the answer would be yes. Then your control strategy would be resolution tested with each system suitability before injection of sample or batch. Now, sometimes you may not have that particular impurity, or it's only available in small quantities. But for example, let's say it's an oxide, and you know you can easily form an N oxide. So what you can do as part of your system suitability, generate a sample, stress sample in situ, right, and run that as part of your system suitability solution. Because one of the worst things to do is when you transfer a method to QC or to a CRO, CMO, and you transfer the method, they're going to ask you for that small amount of that impurity. And if your API organization doesn't have that impurity, it just adds additional labor. But if you think creatively and then make the sample in situ by adding peroxide, then you can avoid that. So now let's move to another example. You have ionic strength of buffer. In this particular case, you have resolution of critical pair. Is the critical method variable, yes or no? If the ionic strength doesn't have an impact on the resolution, for at least this method attribute, it's not critical. So a control strategy would not be needed for ionic strength of the buffer because it didn't have impact of resolution. However, if ionic strength had an impact on another critical method attribute, then you would need a control strategy for that. FMEA, brief overview. I mean, you could do a whole training on FMEA itself, but it is a systematic tool. It relies on method, technique, understanding. It evaluates the method failures and impact on the reportable result. It evaluates the potential causes of failure and their probabilities, and the ability to detect and prevent prior to system suitability, after system suitability, and prior to injection of sample. See, if you can detect it prior to you even running your system suitability sample, that's a great detection mode. If you detect it after system suitability is run, that's okay too. And if you can detect it prior to injection of sample, that's all right. But after your detection mode is only used after the injection of your sample, it's not a really good detection mode. So on a scale of 1 to 5, 5 being the worst, you would get a score of a 5. And the FMEA, what it does is helps to prioritize actions. So let's break it down. Failure mode effect analysis, there's 3 parts. There's probability, impact, and detection. Sometimes impact is called severity. You could use a variety of scales. Most people use 1 to 5, but you can use 1 to 10. So the highest score one can get is, if you're using a 5 scale, 5 times 5 times 5. It's 125. That's very bad. So that would be high risk. The lowest score would be 1 times 1 times 1, which is 1. That's your lowest risk. Each one of these, probability, severity, and detection, have a score, 1 to 5. Probability, the worst score to get is a 5. Severity, the worst score to get is a 5. The same thing with detection. But with detection, the more ways that you have to detect something, okay, a better control strategy, your detection number goes down. Okay, so you want it to go down. Severity or impact, it's a very important note here, regardless of your probability and detection, your impact score will always be the same. You can never change the impact, okay? You can never change the impact or severity. Probability is something that could potentially change, because generally when you do your initial assessments for probability, you may or may not have all the information to make the best informed decision, but you need to put down some type of number and the rationale behind it. As the user starts to gain more information about their product and their method, the probability number could be fine-tuned or tailored. So the best case scenario is 1, and the high-risk scenario would be 125. Now, every company can use their own risk prioritization number ranking, sometimes called RPN. This is just an example. Low risk could be 1 to 9. 10 to 19 could be medium risk, and 20 to 125 could be high risk. Once again, depending on the type of software that you have, knowledge management software, all this can be done in an automated mode. So then once you enter in the specific numbers for your probability, impact, and detection, it color codes it based upon the numbers in the RPN category, and then you have to put in it forces you to put in a reason if it's yellow or red. Here's an example of an output of that type of software. So you have the fishbone. Actually, what happens is you make the fishbone. It automatically transfers everything from the fishbone into the heat map. And it makes it very simple for the end user. End user then just colors it yellow or green or red. And there's a drop-down menu. You create your first DOE, which is a screening DOE based upon the number of variables that might be red or yellow. After the formulation is more or less locked, a synthetic process is more controlled. You then do an FMEA. And the FMEA is a failure mode risk analysis, and you take a look at the probability of something failing, what is the impact if it did fail or moved away from target, and what is the detectability. Then you take a look at all the red and the yellow ones, and then you can set up a DOE2, which is called an optimization DOE. And all of this is already, you have a report, and what it does is as you start to gain pieces of information, it already pre-populates the report template. So at any juncture throughout the development, you just have to hit print, and your report comes out up to whatever stage that you've completed. Very automated. The name of the software, by the way, is Light Pharma. It's a company in Northeast United States. And there's also other softwares that are out there as well, too. But we haven't evaluated them, so I can't really speak on them. Okay, case study, impurity method. So here's an example. In this particular case, this method was already developed and validated. However, there were some challenges. So we ended up trying to use a QBD approach to resolve some of those challenges. Once again, this wasn't the fully proactive approach. This is the reactive approach. But you can still use QBD in a reactive mode, once again, because what is your goal? Your goal is to find out what is the root cause for the failure, and then based upon the root cause, determine what is the control strategy so it doesn't happen again. A fishbone risk analysis was used to evaluate the potential method variables that can impact the CNAs. Statistical design of experiments, the DOE, was also done. The DOE results were reviewed, evaluated, and determined if those method variable effects were statistically significant, looking at both main effects and interactions of the variables on the method attributes. Anything that was deemed critical was called the critical method variable, and then we instilled a control strategy and incorporated that into the method. Here's a fishbone. This is a little bit more comprehensive one. So you take a look at buffer solution. What are all the different things that go into a buffer solution that could deviate from target that you can control? Remember, robustness is something, there are continuous variables, there are things that you could control. Then there's another type of DOE that you could do, which is for ruggedness. Okay, those are things you cannot control day to day, right? Instrument to instrument. Lot number 1 versus lot number 2. Those are things you cannot control. That would be a separate DOE. So you have a robustness DOE, and this is the ruggedness DOE. We're focusing on the robustness one right now. And so you take a look at these, and then a fishbone analysis was done, a heat map analysis was done, and then there were several of these variables that were chosen. And here are the critical method attributes. There's 2 different resolution requirements. There's a quantitation limit requirement. There's a tailing factor requirement, a column efficiency requirement, RSD requirement, and then there's also a requirement of the quantitation of this one impurity. Here's the FMEA risk analysis, and this is called FMEA Light. All right. Once again, this is a very light version of FMEA. You can use this in the initial stages in your organization before a more, let's say, in-depth FMEA program is instilled. But here, this is probability, this is impact detection. If you looked at the prior RPN scores, anything over a certain value got red, anything between 9 and 19 got yellow. But remember, anything that's red or yellow, you need to describe why it's red, why it's yellow. So what's the probability? What's the impact? What's the detection? So we actually have a more elaborate FMEA risk analysis where you actually talk about the probability, you input it. There's something else you need to consider too. It's when you have something, for example, like pH, deviation from target, you need to do 2 risk analysis. One risk analysis, what is the probability that the pH will deviate higher than target? And what's the probability that pH will deviate lower than target? And notice the impact can be different as well for each of those scenarios. Now, the detectability probably will be the same, right, for detecting any deviations in pH. So this is the DOE. A full factorial DOE was used. There's 2 to the n. In this particular case, there's 3 variables, so 2 raised to the power of 3 equals 8 experiments. Typically, you don't just do the 8 experiments. You also need to do replicates. You need to ensure from your replicate analysis what's the normal variability of your method. Also, another thing, a point of note is always run your target experiments up front. Because the worst thing to do is to start running your DOE experiments and find out your instrument's not working, and then you say, oh, the DOE is no good. No, the instrument wasn't working. That's why you want to run it at target first, because you know how the method normally behaves. And then you go ahead and start your DOE. And a lot of times I've seen, doing this for many years now, in multiple companies, and you'll see that people run the DOEs, but they are not running the full system suitability. They just want to get their robustness done quickly. Well, it's something that you need to consider. Remember, as part of your, you need to understand how the system suitability parameters behave in the different robustness conditions you put it in. So you have to run the full system suitability, your bracketing standards, your sensitivity solution, your samples, and then your bracketing standard solutions again for each one of those conditions. All right? And then you can say that you truly vary the conditions and really understand the impact of your method variables on your method attributes. Here's the initial DOE design. This was the initial study sequence. We ran 4, 5, 8, then all low, and then the all high 6 and 7. So 3 targets were run first, passed all system suitability. Then experiments 1 and 2 were run. We ran 1, then we ran 2. When we ran experiment 2, it did not meet the validity and acceptance criteria as single noise for a compound F obtained was 4. The study response criteria was not less than 5. So it indicated, and from this one was an easy one to catch, that the mobile phase composition of 73% was set too high. So what we needed to do here is reevaluate what should be the ranges. So what we ended up doing is changing the high level range from 73% buffer to 72%. Another big challenge or mistake that I've seen over the years is that sometimes people run the DOE but don't check it till 2 days later or 3 days later. No, you need to check it after a couple of key experiments. You run your target, you check it. You also run some of your critical, let's say, experiments, the all high or the all low up front to see if all the parameters, more or less, are generating values within the system suitability, your proposed system suitability criteria. Here's the new revised DOE design. You notice that it's not 73, it's 72. This was run. There's a lot of variety of standard programs you can use. There's Excel, there's JUMP. In this particular case, I think we use JUMP. And you want to take a look at if there's any relationships and significance. Okay, you can have basically trends, but if they're not significant, okay, then they're not critical. So the p-value has to be significant. And not only does it need to be significant, it needs to be of a big magnitude. So for example, just say your resolution, you did see an impact of resolution and it changed from 3 to 3.5 when the pH changed over X value. Well, but that's okay. You don't need to have a control strategy for that because the resolution is so wide that it's not critical. The magnitude is not big enough. Now, on the other hand, you have resolution changing from 1.1 to 1.4. Well, you know what? That's significant and of a big enough magnitude where you need a control strategy. Back to this example. In here, you can clearly see that the buffer concentration is very, very, very critical for the quantitation of this impurity C. So you want to definitely stay away from the higher buffer concentration. You can look at this Pareto chart, and if you look on the left-hand side, what it means is that as you start to increase the buffer strength, what you start to see is a decrease in the response of that variable. That's bad. And in pH, you can see as you increase pH, you see an increase in response of that variable. Notice that the p-value is greater than 0.05, so it's not statistically significant. In this particular case, buffer is statistically significant, less than 0.05, and you can clearly see it from the graph that you can see a significant drop in the quantitation of that as you increase the buffer concentration. Another example, resolution between 2 impurities. Sometimes you get parabolic relationship. These are interaction effects. And in another case, you can also see, look at this column temperature. Once again, it's not significant because it's above 0.05. In this case, buffer concentration is very significant. You have actually a parabolic relationship. So you actually have some target value that you need to work on. Now, you're going to have a lot of different variables, right? And there's a lot of different method attributes. You need to find the overall optimum. So in some cases, you have to say which response are the ones that are going to be the most important. I starred them here. The impurity C, the singular noise, the resolution. And you can see here that obviously the buffer concentration working in this region is no good for this attribute. It's also no good for this attribute here, nor for this one. But then you say, wait a minute, but my plates are higher. But you know what? My plates, okay, it's a method attribute, but I'm way above the system suitability criteria for this particular method. What I really need to focus on is singular noise and the resolution. So then what you need to set is the new target for this method. For column temperature and pH, it really wasn't critical for any of the method variables. It's really buffer concentration. So here's another response surface. You can take a look at it. It's curved. What you really want to see for a really robust method is a flat surface. Here's my knowledge space. This is the region I studied. The operating range, basically the basis for the method operable design region, and then what your set point is. Originally, the method set point was at 70 degrees. Now we moved it to 69%, not 70 degrees, 70%. We moved it to 69%. We know that the instrument variability has a tolerance of plus or minus 2%. So when we have a suitable operating range of 67 to 72, we're within the operating range of the instrument. Obviously, you can never set a control tighter than your instrument variability. So based upon the DOE results, we define the method variables and we define the control strategies. Once again, they're scientifically driven system suitability criteria. This is an example of the overall control strategy. We added a resolution solution and a requirement of not less than 1.1 for this critical pair, impurity C and impurity D. It's included as part of the system suitability requirement, and it's injected prior to the injection of the sample. We optimize the target mobile phase composition. Also, pH is specified as 3.4 in the method and is robust within a certain region. We included some additional preparation details. This wasn't absolutely necessary because it wasn't critical. And this is very, very important. We specified the integration parameters with example chromatograms for all standards. And I want to emphasize again, the same integration must be used for validation as is used for release and stability. Two more slides. This just shows an example of how you could, a revised method with a control strategy. You indicate what is the volume percent of phosphoric acid used. You spell out the formula. You include additional preparation details for how to prepare the buffer and also prepare the mobile phase. As simple as this seems, I can tell you sometimes your pH meter is broken. People are still using Kim wipes on the pH meter. They're using the wrong buffer, whatever it might be. If you do this, if you say with about 0.5 mils and they added 3 mils, something's wrong, right? That means that tells them right there that something is wrong. So this puts an additional control strategy to avoid any particular challenges. This is the overall workflow. I do have a paper on this. If you take a look at LCGC Europe, this is published. But it goes all the way from the quality target profile to your fishbone, traffic risk analysis, FMEA, optimization DOE, control strategy, update the FMEA, and then you can go ahead and do co-validation with your CRO partner or QC. Conclusions, when quality by design is implemented into the development strategy, you truly understand the critical sources of the variability coming from the sample preparation or the analytical method itself. You work together as a team with quality control, your CROs, in order to get the best possible method. You implement the right risk analysis at the right time to understand what are the critical sources of variability, and you control it. You have a really good knowledge management system to document why certain things were changed and what was done to improve the variability due to the parameters or the variables. At the end of the day, when you do this, you can reduce system suitability failures. You'll truly get a return on your investment because you're lowering operating costs, fewer failures and deviation investigations. And at the end of the day, this translates into significant business benefits because you avoid replications. You have better knowledge preservation and greater knowledge exchange and transfer, and you're truly meeting the needs of your customer. Thank you very much.
We have time for one question.
Rosario, nice talk.
Thank you.
Ask several questions, but I guess maybe the one that I want to ask is with regard to, you talked about statistical significance and all your parameters being within that study. Have you looked at maybe setting your ranges wide, not necessarily letting them have them all past what you think is acceptable, and let the practical relevance of that term, i.e. the quantitation, be the driver instead of the resolution?
Yes, in the screening.
Extract that information out of the statistics of the effect size of the model.
Yeah, no, it's a great question. The screening DOE, the first DOE that one would do, you purposely set the parameters wider. For example, temperature, you would set it at plus or minus 7 degrees C, or changing gradient slope from, let's say, 2% to 6%. And there you want to get some failures because you really want to understand where are your boundaries of failure. Okay? But later on, after you're doing your FMEA and you're doing your optimization DOE, there you don't want to get any failures because there you're starting to lock the method down right before your validation. Okay? So in your first DOE, oh yeah, the screening, you really want to understand where the boundaries of failure are. And if you do get failures, that's actually a good thing. Did I answer your question?
Yeah, I still have a question on how
So
you deal
I'm
with
sorry, if you can ask that
statistical
question
versus practical relevance.
just in the
All right.
recess, that would be.
We'll chat after. Thank you again.
Thank you.